bones shouldn't be too heavy. Not much oil in there now.
Roger, thanks.
Ready?
I'm ready.
Let's do it. Let's get some worms.
Whew. So are the ossidacs eating the oil then? Or is it just something that seeps out?
Yeah, they burrow into the whale and they start to break
go.
down the oil and the bacteria metabolize
That was a
it
pretty good
or oxidize it.
chunk there.
Very cool.
Yeah. Here we go. You're becoming a tube worm collecting expert here. All right. Wow. We're going to be knee deep in tube worms. All right. Great. I'm feeling very good at that sample's going to have what we're looking for.
I hope so.
37, Steve?
Correct.
All right. Thank you. All right. So I think next stop, tail.
Next up, tail.
Zooming out.
So Mo, now that you'll eventually receive the humerus bone, do you still need
zoom in a little bit now?
A vertebrae?
I'm going to go past all the junk.
Yeah, go past the junk. There you go.
We're also curious to hear if anyone knows what the yellow is that we're seeing in between the vertebrae.
My vote is bacterial, Matt. Yellow. So, Jen, Mo, now that the humerus has been collected, and it'll probably go to the worm biologist first, and then it can go to Cal Academy. Cal Academy does not necessarily need a vertebrae. So the vertebrae collection could be for the worms.
Okay.
And if after they're done with that vertebrae,
if they have to preserve the worms in the bone, does that make a difference? Ultimately,
ultimately no, because they'll take the bones just frozen in the -80. And then Greg Rouse is going to be on the ship on the last cruise, so we don't even need to take them off. And then they'll extract them in the lab, and then the bone will still be there.
All right, so where are we going on the tail here?
So now we're looking then for a vertebrae that's small but has ossidacs. Of the 4 that are loose, the one on the left seems the fuzziest to me.
Bob, is everybody level good to you? Audio level? Anybody low or high?
Talking to me?
Yeah.
Okay.
Good.
Oh, and Jen, I missed this part. They could also, Cal Academy could also archive the ossidacs. So a bone with the ossidacs for Cal Academy that's not going to, where the ossidacs is not going to be removed, could also go to Cal Academy. So any of these smaller bones would be fine too.
Okay.
That's a bonus.
And I don't know if they necessarily need to take, say the humerus had a lot of worms in it. Maybe they would leave some behind for archiving, but I guess we'll have to just see how many they need and what they can leave behind.
Yeah, I mean, if it's easy and it's small, one of these smaller vertebrae could go to Cal Academy with the ossidacs on it, and then another one can go to ossidacs biologist.
Per the instructions, I think the one that they're most likely to be interested in, probably that mandible piece, because it's very friable, which is what they wrote in their protocol.
They wanted friable bone.
Yeah. But it did look like there was 2 species of ossidacs on the humerus.
Yeah,
Hey, science team, this is Amity. I'm sure our audience would love to get a zoom in on an Ossidax when we've got good viz and we have a little time.
We had some really
Some
good.
folks have never seen it up close before.
We had
It's
some
pretty
really
cool.
good zooms, yeah, like 10 minutes ago, of a couple of different morphs, potentially 2 different species that were side by side. They were beautiful.
Mm-hmm.
Yeah.
So
And these are all the females.
Right.
What are
I
the
think
females?
for most species of Oxidax, the large worm that you see visible are females. Males are very small and
small and like parasitic.
Right. Though I thought I read there's at least one species where the male is large. But what we're looking at is the kind of carpet or furry structures on top of these bones that are pinkish. And if you look closely, they're in a tube that's going into the bone. So when the water clears here, we're going to zoom in on some of these vertebrae near the tail of where the tail would have been on this whale. And we're looking to see if there are any Oxidax present.
Looks like there's some on the 3 that are really close together.
Yeah.
That are now out of frame. Oh, yeah.
The one that's in the bottom of the 3 has a lot of
You got
worm
more
on
zoom
it.
there, Al?
And it looks pretty friable also.
It does.
We'll have to see if it holds up to collection. But if it did, that's probably a great selection for worm extraction.
Agree.
Okay, so let's try that
Okay.
first. Could we put it in with the baleen so that we don't have to carry it as far?
If it's soft and friable, we can probably put it in there, but let's try, for starboard E first if we can get it there.
Okay.
If it starts to fall apart, we'll have a contingency.
Okay, zoom out.
That is if we're not going to pick up any more larger bones after this, because that's our last big box. Any thoughts?
That's not a very big bone there. I mean, judging by the lasers, right? Like
10 centimeters?
Yeah, I'm just concerned that it might not fit fully in the slot on the starboard inner, but we can take a look.
Okay.
Yeah, I don't think we need any large bones after this one.
And if we have time, the auditory bula, it
true.
could take some time to extract that, but that's less than 10 those are less than 10 centimeters.
Okay.
So those are those just normally grabbed and pulled off, or how does that
go
work?
Go ahead and zoom there.
I don't know.
I've
never interacted with a dead whale before this one.
You haven't? What do you?
Yeah, this is
Everybody's
my first whale.
done this. Come on.
There was a message in the chat a while back that they used a hammer.
A hammer.
Yeah. So we've got an arm, hydraulic arm.
Yeah.
Oh, jeez.
And that's on a much fresher specimen on the beach, where you have to cut away all the tissue. I wouldn't say fresh, but a lot fresher than this.
I don't think it's that fragile.
Okay.
Let's go starboard.
Great. Can we rotate it before we
let's get some images.
Transport?
Yeah.
Very nice.
Wow, Crispin Little says Ossidax borings into whale and other vertebrae bones are quite distinctive and have been recognized in the fossil record. Wow.
This is a very long association that we see it in fossils as well. Very interesting.
Porous.
There's a little amphipod on there, some amphipods it looks like.
Okay.
All right, thank you.
Beautiful.
Okay, zoom out.
Is there a name for this bone specifically?
Caudal vertebrae.
Okay.
Caudal means tail.
Yeah, CAU.
DAL. For anyone who's
DAL.
curious.
Ready for
38, Steve?
That is right. Yep. Yeah, I guess if you can go in one of the inner boxes, Bob, that would be ideal. You pick, they're all empty.
They're all open?
Yeah.
Okay.
If any of the audience members are on the quad cam view, if you want to look at camera 3, stream 3, I think you can see where the sample is being placed.
And the boxes have letter labels visible in that camera view.
That go in C or
Hey,
D?
Jen, we have a couple.
I saw D.
I went D.
See.
D.
D for dog.
A couple of questions from the audience. Do we have any sense about how these Osidax find the whale falls? How do they transport between one or the other?
That's a great question. So, you know, besides the live or the natural whale falls, they've done a lot of experiments with whales that were beached, like Rosebud, that Mbari towed out and sank, and they studied that for, you know, year after year, to alligator carcasses, to cows, to all sorts of vertebrates they just tossed in the water. And every single one of these ones or animals is colonized. So their larvae are just ubiquitous in the world's, most of the world's oceans, if not all the world's oceans. I don't know the mechanism in which the larvae touches it or senses the bone and locks on, but they are pretty cosmopolitan in the world's oceans. There's 30-something species or so, and they only exist on bones. It's amazing, because oh, sorry, I had shore going there.
Okay, what are we doing?
We are discussing really quickly over on the Nautilus line. Hold 1 second.
Want to zoom in? Out. I don't like this spot.
I'm zooming out.
Yeah, every dust storm has happened on this back corner.
Yeah, because it's a little bit, it's upslope right there. There's a little mound, you know?
Yeah.
So if you try and set down all the back of the vehicles there, and you get the current coming from our back, it's like blowing it over us.
Yeah.
Wafting over.
I'm going to go back in now.
All right. Yeah, the worst is when you get like a slope, a muddy slope, and you set down next to it with the horizontal.
Oh, yeah.
It just, ooh.
Yeah, it only takes a few seconds to make a big mess, and then it takes a long time to settle back out.
Yeah. And if you set up with the current in your back, it comes around, and then it makes a vortex in the front, so it kind of just swirls the mud around in front of you, you know?
Yeah. I've seen the vortexes like off on the side a few times on these dives, but
Okay, so we have a plan, and that plan is, as soon as this clears up, to lift off and travel up the left side of the whale, which is now the opposite side that we traveled down.
Okay.
Look for anything interesting to zoom in on. I would definitely like to get some zooms in on the yellow in between the vertebrae.
Okay.
And anything else that might catch our eye. And then we are going to take a poke at those auditory ambuli and see whether there's an opportunity to remove one.
Okay.
Take it with us.
We're getting yanked on?
Yeah, we're pretty far away, and the ship is moving more.
Yeah, we got a little bit of
we gotta
wind.
clean off the dust here.
I
Gotta clean your windows.
I can come down a little bit.
Yeah. Come down, and Sam, can you move us over there?
Yep, sure can.
Does that fine silt just stick to the vehicle from
Yeah.
the weight?
Yeah. Whenever you sit down, you pick up dirt.
Yeah.
Did you
And then
budget for
it's
that
with
in your trip to the surface?
Paul, your heading is 160 or
Mud sticking to your shoes or something.
150?
Yeah, I'm about 145. Oh, no, yeah.
In blue, right?
Pardon?
It's in blue right there, right above your tension. Is that your heading?
Yeah. Sometimes I look more at the like reference and goal.
Okay, I'm gonna move
that's where I'm
ship east.
trying to be, so it's working its way.
I wish I was
Bridge
young enough
nav.
to see that. I can see it.
Actually, slight space. 10 meters, 112.
So Amity, I know we haven't had a lot
little
of time
southeast.
in between science and operations to answer questions, but once we're done here, we are going to have 3+ hours of blue water. So we'll have time to answer some questions then.
Yeah,
everyone
absolutely.
to
I've
know
been
that
holding off, just trying to give way to the science. Good observations.
We also have a wrap, so I can do almost a full spin. Should I go for it?
We're kind of low. I don't know.
All right.
Hey, Jan, we are getting a question. Do you think you're going to grab a sample of the yellow, I guess it's a vertebrate? With
Well,
the
there's also a
potential
wrap in the 6.8, but I guess we have no idea.
The yellow, I think the yellow is new to the whale fall, at least new as it wasn't there last year. Correct me if I'm wrong, Chad or
I'll just take
Jen,
my wrap
but
out.
I don't see
It's going to help get the dust off.
for that. Frankly, I don't really know what it is.
I guess the question is, are you gonna sample? You grab a sample of that?
I don't think so. Yeah.
No.
I think we can definitely get some tight shots of it and then maybe pass it to some experts for their best educated guess. But yeah, we were discussing
we don't have any requests for it.
Yeah, we don't have any requests for it and wouldn't know exactly who to give it to right away anyway. But the other thing is, even though we have a permit to collect more bones, we also want to leave as many bones here as we can. So we've fulfilled requests.
Should I start to come up a little bit now that we're getting Argus's? Yeah.
Too much.
And that ship move is about to complete. Do we want to take another step?
Yeah.
Roger. Bridge nav. 10 meters, 112.
Hey, Jan, we have a question here about, do we know anything about the sinking rate of whales? Like how long would it take a whale on the surface to reach this depth once it dies?
Once it starts sinking, that's a good question. I don't really know the answer. Like we can speculate. Different species of whales will sink or float depending on how they're killed. Are they killed by a predator like a killer whale, which will create holes in the body? And so holes in the body will release the decomposition gases more quickly. And so it's going to sink more quickly. So getting rid of the gases and the blubber. So some carcasses are fed upon by things like white sharks, and they will just flinch off. They'll take their teeth and just kind of just tear right into and kind of flinch off the blubber. Not necessarily eating the meat as far as the white sharks are concerned. They really want that rich, fatty blubber. Those whales will sink a lot faster.
On the yellow to the right of the lasers anymore?
Looks like, there you go. Thanks, Bob. I'm going in.
And then some whales will sink right away, just naturally. Ooh. And then they'll refloat.
Kind of yanking on me.
So
there we go.
The answer is really,
it
depends.
Up, but not that much.
Yeah.
So we're thinking that yellow might be microbial mat. And input from Crispin Little, a scientist ashore, is that they can be very different colors. So yellow is one color that microbial mats can be. And that's a really nice view. Thank you.
Ship move complete. Want another step?
Yeah, I guess so.
Roger.
Yeah.
Bridge nav.
There are a lot of amphipods on the bone, and you can see how porous the bone is as well.
10 meters, 157.
That's good, thank you.
Okay, zoom
Coming out, Bob.
Yeah, it's interesting that the bacterial mat is on the inner part of the vertebrae.
Can you say that again, Amity?
A little bit.
Oh, just noticing the bacteria is on the kind of the inner centric part of the vertebrae, not on the outer.
Yeah, so the last year that was all full of cartilage still, those spaces between the vertebrae. And that's gone. And in some, I don't see it in all of these locations where the cartilage was this yellow, but it's definitely in a number of them. I don't know if it's the remaining cartilage that they're eating or something on the interior of the bone.
Yeah.
Or I should say that's supporting the yellow.
The disc plates that are starting to separate from the vertebrae. That's bone on top of bone. Oh, we can see some of the little phalanges.
Can you describe that where you're looking?
But on the right. Oh, just went out of screen, and then there was another one further to the right.
Below
that
this
one
skeleton?
And that one.
Oh.
Yeah. The phalanges are part of your fingers.
Where?
Upper screen just went out over the top. There's one on the
top of the screen.
right.
Oh, I see.
There you go.
Right, top right screen. In the top of frame. And if you pan
definitely
right
that farthest right one, and then
okay.
I'm suspecting that the middle and the left one are also phalanges.
Zoom in on that.
The farthest right one is definitely a phalange.
And I'm assuming they're this far away from where we saw the humerus and the other arm bones because scavengers who were
Zoom out.
feeding on them have dragged them over to these locations.
Thing?
Oh, I think that's the benthic ctenophore that Amanda was pointing out earlier.
Oh, yeah.
Great, thank you.
Yeah, just and they're scattered
zoom
just
on the
like
other one?
baleen plates were scattered. Scavengers taking them all apart.
Want to zoom in now?
Looks like these flat structures on the seafloor are those projections that were on the vertebrae that have now fallen off. And
yeah, you got like a top process. If you feel your backbone, you'll feel the top, the spinous process. And then they have like transverse processes coming off the sides.
Okay, zoom out now.
Oh, there's a couple of them. In between the two vertebrae, I see them now. I was looking at in the lower left, but there's a number of... You're thinking those are scale worms, Steve?
Yeah. I wasn't sure if they were isopods or worms. Sometimes isopods
I'll get it over there.
Flat scales like that, but they're probably worms.
Those are the largest scale worms I've ever seen. They're just tiny, like a less than half of a centimeter.
Down in the kind of lower left area.
All right, zoom in.
Are we looking at another phalange or at the scale worms? Aha, there we go. Well, Amity, here are those other ossidacs that we were mentioning at the top of the shot that have a much thicker tube, wider tube, I should say, and are taller and more red in coloration. So we're thinking there's at least two species here currently.
Okay.
Or is that male versus female?
That's not my understanding.
Can you zoom in more out?
That's into my lens.
My understanding is
the male. You don't even see the males. Yeah, I think most species, as Chad mentioned,
really small.
Yeah,
they're basically stunted larvae. I think they don't really mature into adult stages.
Okay, so these most likely are at least two species of ossidacs.
Yeah,
the males are mostly there to produce sperm.
We last year there were different color morphs, but the same structure. There were some yellow, orange, and red, and a lot of those actually ended up being the same species, just different, polymorphs. But these two are really distinct. So I would say they're two different.
Okay, zoom out.
All right. Let's keep.
Yeah,
that's great. Thank you for
the
zoom.
If I recall
that
from
maybe that
last year,
yellow is just decomposing cartilage that's left on the bone. Because if you look where the scale worms were feeding, it looks like they're kind of cleaning off that yellow. I don't know. I've never seen cartilage look that color, but I've never seen a whale fall before.
Well, and it could be the yellow is bacteria on cartilage, and scale worms could be eating either one. So, interesting.
Can we bump the camera up just a little bit? I don't know if it'll zoom out, but to get those ossidacs more in full view. Yeah.
Pan up.
There you go.
There we go. Great,
nice.
Thank you.
Oh, that's beautiful.
All right, thank you.
Science time check, we've got about a half hour before we need to start ascent.
Okay.
Yep, so we should probably pick up and get on over to
Gile?
Yes.
Yeah.
I think that's where we'll head next. These were great shots, though, to look at how many different types of animals are currently inhabiting and making their living off this whale.
Okay, zoom out now.
If you look really closely, there's tons of little white crustaceans flittering about. Almost look like flea size.
Past the junk.
All right.
There you go.
Yeah, I was just going to add that I think Amanda last year had said that a lot of the typically large females of the Osidax actually, they hold dozens of males near them. They just kind of ready to accept the sperm when their eggs are, to fertilize their eggs. So I think Chad had mentioned that they were hanging around, so to speak.
Yeah. So Bob, when you're ready, we're ready to head towards the head.
Okay.
Thank you. As we fly that way, we're going to pass over we're going to go past a sea spider. A pycnogonid. It's going to pass under the lasers. It's not on the whale. It's right below the whale.
The lasers are almost right above it now. Oh, there's a couple of them. Oh, there's several.
What are these gelatinous
What are we looking at?
shapes?
Zoom in?
Spiders.
Large sea spiders.
And what's the gelatinous shape underneath the spine
The
there?
bottom right frame.
That is hopefully a question
one
someone
there.
will help us with. But the pycnogonid, yeah, is on the right side.
Never mind.
And now there's one in the center. See, there's more.
And I know there was one on
On the right.
one of the auditory bullae. We got
Yeah,
a nice look
maybe
at it earlier.
There it is.
Yep.
There's one.
Ooh, spiders materializing out of the mist.
It's almost Halloween.
Oh, they're eating something. They
There
have
we
their
go.
proboscis extended. Maybe they're preying on the worms.
Hmm.
You can see the proboscis right here. Here.
All right.
Extended
I just want to keep.
into
Hey, zoom
worm
out.
tube or something.
I love taking these looks, but also want to keep moving. I'm torn.
Torn.
Steve, we're going to take a collection and see if we can get you a telestrator for Christmas.
You know I would use it too. That's the crazy part. You're going to have to figure out how to pipe it into the van from my house.
You could be our test bed for telestrators for everyone at home.
All right.
What do those go for? I'm just
We still-
curious.
I've never purchased one,
Are
but
we
I
moving?
understand they're kind of
Or
pricey.
I delayed.
Maybe you don't need that fancy one. I mean, seems like just a webcam drawn on the screen kind of thing.
I was going to say, up until this year, we had the stick that we just used.
Yeah.
You can use a laser pointer.
That's what
pointing?
I'm used to using.
Pardon?
The big
Oh, I
stick.
thought you were pointing at something.
Oh, no.
We can go old school. We can go overhead projector on screen.
Now we're moving up the whale. We're going to start encountering some ribs laying on the sediments, both above and below. A little eel pout fish poking out, looking out from between the ribs. And white squat lobster are also larger ones. We've also, when we zoomed in, seen smaller ones. Another eel pout. And there, the pycnogonid, the sea spider, is still on that auditory ambulae. Must be a nice spot.
That sea spider's still there. So I was thinking this lower one is going to be a little bit easier.
Want to zoom in now?
And why do you think that one's easier, Jan?
It looked like it had the bone that it sits on is cracked, so it might be able to be just lifted off.
Okay.
Maybe.
We can hope it'd be that easy.
So we going
Hey,
for
Jan,
that?
can
Will you be able to
explain
reach it?
what that is for the
Yes.
audience when you get a sec?
Okay, zoom in
Will-
out there, Al. Sorry.
Bob, do you think you'll be able to reach
So the
that?
auditory bullae hold our
Sorry,
ear
zoom out,
bones.
Al.
And in marine mammals, because they dive so deep, the outer portion of the bone itself is pretty dense material, bony material. And it protects all of those very delicate, you know, the stapes. What are the other two bones?
Oh, the inner
Help
ear
me out
bones?
here.
Hammer.
Yeah, the inner ear bones.
And
There you go. The hammer, the anvil, stapes.
Steve, I texted Ed. He said around 30 grand.
Oh, chump change.
For some people.
30 grand, just like AirPlay or iPad.
Yeah,
you know?
so we can trade a day of ship time for one
webcam in a house.
Yeah.
one screen.
Microsoft Paint.
I think there's got to be a middle ground between those options.
we have a grenadier fish making an appearance here.
Zoom out.
My wife and kids just logged on this morning, so I want to say hi to them.
Can we get some close-ups before we go in with the claw on, with lasers on that bone?
Hi Maya, hi Noah. Be nice to mom.
And off.
Good capture. Yeah, lasers off.
Get more zoom now.
Zoom.
All right. Ready to go.
All right.
Here we go.
Did we get one with lasers? Sorry,
Yes.
I was typing. Okay, thanks. The grenadier is very interested to watch. Don't let it make you nervous, okay?
Are we slurping?
Oh, whoops. What am I doing?
No, I think you have to grab it.
Slurping? No, I
I
think
don't know why I was doing
Okay.
I don't think that's going to be slurping.
See,
Nope.
He's gone
it was the
rogue.
grenadier, right?
Kind of looks like
The
a sponge.
grenadier made you nervous, but
Grenadier
it's okay.
supervising.
Okay, zoom in now.
Been sampling corals and sponges for too long. Everything looks like it's slurpable.
That's a pretty hard bone, so
Okay, but maybe not.
I don't know how friable it can become.
We're about to find out.
Making contact in 3, 2, and 1. Oh, and
1.
1.
Oh, I think you
Go.
got it.
Oh!
Now if you can get it back without dropping.
Wow.
Beautiful.
This is one of those
Perfect.
moments where the delay, I heard your happy sounds before I saw it.
Oh, there you go.
Yeah, spoilers.
That was really good.
Can we do the carousel?
Okay.
It's very much like the
Let's
claw
see.
game.
Let's bring it down a little
so
bit.
this is from Mo. It might be possible to age it with the ear bone, but not necessarily. If there were an earplug, that would work better. But I'm sure that's long gone.
There we go.
Wow.
I'm
There's-
impressed too. Mo says, I'm impressed.
Wow.
Oh,
Shot
yeah,
reminds me
that.
of
It did look a little loose, that one. The other one looked a little bit harder. Oh, that's nice.
Wow, very cool.
Where is this one going to go,
What do you think of the
Steve?
inside of that, Mo?
You got a good grip on this one, Bob?
I do.
All right, let's go starboard box in C.
Okay.
B or C.
ID number, Steve?
This is going to be 039.
Roger.
And again, if you want to see the bone go all the way into the collection boxes, look at channel 3.
Ready for the tryout?
Ready.
If this is going to be the last one, we can actually even just put it in E. There's nothing in E. So might as well go. You can go in the big one in E. Just easier. Save us some couple crucial seconds. All right, it's good collection.
Sample tray in.
The ear bone's connected to the which bone? I guess the ear bone's connected to the which bone. Which bone is it connected to, though?
I believe the whale tail.
A whale of a tail. No, legitimate question. What is it? I mean, is it attached to? We just saw where it came from. But what's that bone called?
Head bone, the skull. I was just waiting
it
for
is the skull. Okay.
someone on
Just
Science Chat to
at-
help out. This view is really
Sorry, I thought
awesome,
my
isn't it?
microphone was on. Yeah, it's attached to the skull. just like
remember, the skull-
we have Bule also.
And remember, this whale is laying on its back, so we're looking. That's the underside of the skull. So they don't have these on the top of their skull, but it looks like it would be behind where the jaw attached.
Gotcha.
To the skull.
Yeah, I keep forgetting that's upside down.
Is that right, Jan?
Yeah. Well, you have.
Yeah, the jaw's a bit farther forward.
Mm-hmm. Okay.
All right,
It's kind of tucked in and underneath the skull.
You can spin it out.
Do we, Chad, this is more of a question for you. Do we want the other auditory bule, or do you want to leave it in place?
I think we can leave that in place. I mean, we don't have a need for it, so
Yeah, I don't.
again, try to take as little as we need.
I don't know.
Yeah, I brought it up just because it was
Look
so
like it
easy
would
to
be.
get.
Worst case.
All right. Well, according to my list of what we were hoping to accomplish here at the whale fall, I've checked all the boxes.
That looks like
So
the
that's
long
pretty
way.
exciting.
I think so.
And I have to say, this shot right here is really impressive. Looking at the head of this whale laying on its back with scattered
Why don't you
baleen.
just take it clockwise then a little ways and see if it looks better. Do a half.
Pilots, do you need time to sort out that wrap, or?
We can. Well, we're doing it as we are doing our thing here. But
Okay.
Yeah.
We've got 15 minutes. Is that time that we're gonna need for the wrap, or can we use it for last minute science?
We can do science. He's gonna try and.
Okay.
So we can do some Argus beauty shots if you think you can.
Oh,
that
well, that's the one thing we can't do. With
Oh. Okay.
We're resetting.
Any water needed here? Watch leads?
Bring it around and then.
Or
looks like.
any reason? Niskins?
Didn't have any specific.
I didn't hear
requests
the
for
question.
Any water needed for Niskin
Niskins.
bottles?
There wasn't anything specific to the whale fall. That had been requested.
eDNA.
But you want to do eDNA for the whale? And I guess all these.
Yeah, you can do
species, right,
eDNA
over
for
here.
all of the worms and for the whale.
Yeah, we can do that. You want to take a water sample?
I think so.
Okay.
Jen?
Sorry, I was reading something incoming.
Okay.
Yeah, for the eDNA, how close to, is this close enough?
I think it's plenty close, yeah.
Okay. So then,
Okay.
yeah, we can,
Niskin?
let's
Yeah.
fire Niskin.
Yeah, let's fire Niskin. We don't need any cores, I don't believe. Good there. And for those of you
I mean,
watching,
unless Taylor Anne wanted a core further away from the whale, and we had time to get it. But I know she's going to get cores down in, or she'll be getting cores for sediments down in Channel Islands.
Does she have a single core yet?
We have not taken a core for her.
No, not for microplastics. Here. We have a core for the particulate organic matter research that was going on for the bamboo corals. Can we pop,
that was in Pioneer Canyon.
Can we pop 2 here? Just maybe 1 and 2, just so we have extra water. I don't think we're going to need to sample all Nixons here. So.
You want to put the Neskin rack up on the video? Okay, ready? Here we go.
Yo, pop 1.
Did it go? I think it did. It's hard to see that one. It's way in the back.
It's the farthest one in the back, right? Yeah, it's vertical.
Okay.
Hey, Jen?
Yes.
What is eDNA and why do we want to collect it?
eDNA stands for environmental DNA, and one way to think about that is that all living things are shedding cells and components of their body parts and their waste.
2 bottles.
We shed hair.
2 bottles?
Yep.
Check.
We shed skin. All these animals are also shedding skin cells. They're excreting waste, and all that contains their DNA, and it's present and in the water around them. And so if you collect water, you can look to see what DNA is in that water sample. And if you have also taken these like visual recordings of what is in the area, you can compare the two.
Need time to sort this out.
Roger. Science, we're going to need to take a few minutes to sort out this tether issue before we start ascent.
Okay.
Okay.
Roger that.
Okay.
Perfect.
That was sample 40 for the waypoint.
41?
40.
40. And do you label that differently than others, or just same NA122 40?
40, yeah.
Thanks.
So
and
this is
Steve, you're going to combine 1 and 2 into one sample?
Clockwise spin.
Yeah. So yeah, we're just taking extra water here, for whatever is needed for microplastics. They need about 2 liters, and eDNA needs about 1 liter. And then we also have a request from USGS for doing particulate organic matter. When they can use as much water as possible, and particularly more if there's less POM in the water, because of you need more to filter more water to get more material in the filter. So
okay, so and these were all requests at the seafloor?
Yep. That's right.
Do any of those want water on our ascent?
No really direct measurements or requests, but maybe we can do like 100 meters if we're on schedule. Between 50 and 100 meters for microplastics. Taylor Ann had requested more resolution of sampling in that area around the picnic line, mixed layer area. So I'll put down a note. She's coming on watch next, so she can direct that. And yeah, probably within the 50 to 100 meter range.
Just real quickly, I wanted to, for anyone watching out there on the internet, we're going to wave goodbye to the whale fall. Bye, Mr. Whale.
Bye, whale fall.
Maybe Miss
Bye,
Whale. I don't know.
Come down.
We'll see you next year.
Maybe not. Decomposition's happening really fast.
Yeah, might not be much left. So that kind of concludes the science portion of this dive, which we launched at 4 p.m. Pacific time yesterday. So we've been in the water for roughly 20 hours, doing about 16 hours of science. We went to the octo cone for the majority of this dive. It was a fantastic dive, great scenery, so much wonderful science done.
Yeah, okay, all that looks good, right?
Okay.
And all of these videos can be found on the Nautilus YouTube page at some point.
Yeah, I think so. So right now we're facing each other with -1 wrap.
Yeah.
But then it says that there's 2 wraps in the 6-8, but we reset that, so who knows?
Well, you should come around to be on the heading of the ship, and then, but I got to get closer to you to be able to do that.
And then just for schedule-wise, we're going to be ascending soon on deck, so there's a lot of blue
up
water
a
if you
little
want
bit.
to look at that.
Yeah.
But we launch our next dive approximately 8 p.m. Pacific time tonight at the Octopus Garden. Now it will be a 3-hour descent, so we won't see the octopus till 11 o'clock p.m. Pacific or so, but we'll be collecting some scientific equipment we left out there a year ago that's measuring the temperature, oxygen, and some of the water chemistry. So that'll be a relatively short dive.
Close and then see what it does.
Overnight, and then we'll be done by 8
Yeah.
a.m. Sunday morning. And then we may not be diving for a day or two, as we may have to move out of this Central Coast area for a fairly significant weather system.
I'm just going to hold steady here and let you.
That's moving in. And we may have to move down to the Channel Islands, but hopefully we'll come back up here in a few days to do a dive on an unexplored portion off the base of Davidson Seamount to hopefully find coral sponges, and if we're lucky, more warm water seeps and octopus nurseries.
So I was going to get
Thanks
rid
for
of
that summary, Chad. That was great.
Go this way.
Quick question, Ken. I heard you say that 8 o'clock is when we're thinking of having ROVs in the water tonight, right?
8 o'clock p.m. is when we're going to be launching tonight.
Okay. Just checking because I'm on 8 to 12, so just making a mental note of that.
You get the chalet shift?
Yes, we will shepherd you to the seafloor.
Yes.
Well, I guess Erica, no, who's coming on next? Yeah, Erica and Kirsten to the seafloor.
Okay.
Yeah, now they get to shepherd,
recovery heading?
they get to chalet up.
Yeah, and I'll stay on as well. So if there are questions, I know we've got some questions coming in over the last hour. Haven't been able to get to too many of them just because we had a lot of information going on, a lot of direction and science.
To counter.
Which way are you going to turn Argus?
Which way do we want to be pointing?
To take if there's any wraps in the tether, which way do we want to turn Argus? Argus is currently looking at me. It needs to turn 180.
I think we want to go clockwise. I think we want to be at -1. No, okay. Counterclockwise? I don't think we want to go to -2. Yeah.
Yeah, go up some.
Yep.
See.
On the video or winch?
Just pull up on Argus.
Well, 5 minutes to the watch change. Feel a little bit bad that we had the previous watch get us there, and then we enjoy the entire whale fall, and now we're gone.
Oh, that's true. You guys came on board and then got to the whale fall within 20, 30 minutes or whatever it was, huh?
Sorry, what was that, Bob?
I wanted to look at it more.
Oh, I can come back around?
Yeah.
Nice Tina for.
Which camera? Which camera? Yeah, I don't think I can control it from this computer. Oh, no, I can do it. I can do it.
Are you still trying to come around or what? What's happening?
Yeah.
don't think we're not really gaining anything here. I mean, we're not any information.
Yeah.
Trying to figure out which way to go here. No, not, never mind that. It's a what? Oh, we had reset, so all bets are off.
Twice, actually.
Yeah, all bets are off. We got to go by what it looks like and not by what we see count-wise.
We gotta go by what it looks like and not what we see.
Exactly.
Uh-huh.
Yeah.
So we are in the middle of a shift
I'm
change,
gonna
and the
come up and
pilots.
back and just try and look at it. I'm gonna come up and back. You just stay put, and then we'll look and see what it does. You're gonna have to look up and down. But
So the ROV pilots are resolving a tether issue. We'll be back on in a little bit.
is that all the down we got? All right, I'll come up again. See what we're doing. Yeah. with Herc. Well, let me do one.